DNA is a very large molecule that consists of two strands twisted into a double helix held together by a simple pairing rule. A pairs with T and G pairs with C. It's made up of 3 billion letters or bases, which holds the code to 20,000 genes. Our genes shape who we are as individuals and as a species. Genes also have a profound effect on our health. Researchers are looking for ways to control and manipulate genes, but changing genes in living cells is not an easy task, until now. A new genetic engineering technique has been discovered which promises to dramatically improve our ability to edit genes in all living organisms, including humans. I sat down with Dr. Vardit Ravistky to learn more about this new technology and the ethics that surround it. CRISPR was named the 2015 Technology of the Year. Can you explain briefly what CRISPR means?
CRISPR is an acronym that means clustered, regularly interspaced, short palindromic repeats. Now, what does that actually mean? DNA is actually a series of letters, base pairs, letters. So CRISPR has been compared by everybody to a word processor. It's a tool that allows us to edit the genome, to edit those letters. How does it do that? It cuts the DNA in a specific point that we told it where to cut, and then it replaces the gene that we cut out with the gene that we want to insert into the genome. So for example, if a person has a mutation in a specific gene that can cause disease, CRISPR can get into the cell, cut out that disease gene, that gene that is responsible for disease, and replace it with the correct gene. And we hope that this will result in a healthy person. How does that happen? The CRISPR system is composed of 3 components. There's a guide RNA that tells the system where to cut. Then there are proteins, and that's the Cas part of the system that actually do the cutting. And then there's replacement DNA that once we cut and we have a gap, an opening in the DNA, will insert the correct gene and paste it. So it's really like a word processor. If you wrote an essay for school and you have a mistake, you want to cut out a word, you would use in Word the function of cut, and then you want to paste in the correct word, that would be the Cas system that sort of pastes the correct gene into the DNA. CRISPR allows scientists to do this in a way that is precise, efficient, and not very expensive. It comes as a kit that includes all those 3 components: the target, where are we going to cut, the proteins that cut, and the replacement DNA. So it really allows scientists who work in any area of biology, plant biology, animal biology, and even human genetics, to use this precise and efficient tool to edit genes and to modify the genome.
Now, this is definitely a great discovery, and I understand that scientists are using it in plants and animals, but it's not being used in humans yet because there are ethical issues related to that. Can you maybe explain a bit of the ethics behind the use of CRISPR in humans?
So there are 2 types of uses in human beings. One is to treat patients who suffer from a genetic disease. For example, you and I could be carrier of a gene, a mutation that causes us some devastating disease like Huntington or cystic fibrosis. And we would be candidates for CRISPR therapy in the sense that clinicians would inject CRISPR into our cells, and the CRISPR would go after the bad gene, cut it out, and replace it with a healthy gene. What are the ethical issues with that? As in any other novel therapy, there are risks that we're unaware of, while there are great benefits. Some of those genetic diseases we don't have a cure for. So CRISPR is, right now, the only hope for a cure. Imagine a parent of a sick child knowing there's no cure and reading about CRISPR, thinking this is the last hope for my child. Scientists are currently getting phone calls from desperate parents offering to enroll their children in such clinical trials because they're so desperately looking for a solution. So I think we're not going to have a problem identifying candidates for clinical trials, but making sure those trials are safe enough for human beings is a huge ethical challenge. How do you assess the risks when you've never done it before, while the pressure is on to start the trial because people are so anxious to benefit from what the technology has to offer? So you have issues of designing clinical trials in a safe way and consent. How do you get consent from participants in those trials when this is the first time we're doing it? We've never done it before. How do you give them information when you yourself as a scientist don't know what's going to happen? We keep saying that CRISPR is precise, that the Cas knows where to cut because we gave it a target. But sometimes CRISPR edits in the wrong place. It's called off-target editing. If this happens in a plant, okay, you didn't get the outcome you wanted. You toss it away and start again. If this happens to your child, and where CRISPR cut is actually a healthy gene that your child needs in order to breathe or to see, of course, this is a risk nobody's willing to take. So everybody agrees that for now, we have to keep trying it out on plants and animals until it's precise enough that we have minimized this risk of off-target editing, so that when we introduce it into patients, like adults, Yakir and I, or sick children, it would do a better job of editing exactly where we want it to edit. Treating individuals with CRISPR is challenging because in order to cut out the bad gene, the disease gene, we would have to fix it in all your cells, and you have millions of those. But if we treated an embryo before it started developing and it's just one cell, we could just cut and paste one time in one cell, and then this cell starts to duplicate and replicate, and the entire embryo and then fetus and then child that would develop out of this embryo would have the correct gene. So it's more like preventing the disease before the human being even started developing. This is extremely attractive. It seems like a much simpler solution to genetic disease to just cut and paste and correct before we even developed into human beings. But what is the problem? If I edit your genes, I'm curing you, but I'm not changing in any way your children. If I edit a human embryo, this embryo will now develop with a different genome that we, as scientists, created, that we edited. This edit, this change that we made, will be passed on to this person's children and grandchildren and to all the descendants of that individual. So in fact, if by modifying one embryo, we could potentially be modifying millions of human beings if we look at the evolutionary scale. Because one person can have 2 children who will then have 4 children each, who will then, it's exponential growth. So by editing the genome of one embryo, we could potentially be revolutionizing the direction of human evolution. This is very scary. So when you think about using CRISPR to edit the genome of human embryos, the benefits are enormous. We could potentially sequence the entire genome of a new embryo, identify all the bad mutations, cut them out, replace them with healthy genes, and bring into the world a completely healthy child. Of course, it will never be possible to have completely healthy children, but we could get rid of the most devastating genetic diseases at the level of the embryo. So it's extremely attractive. It would be the ultimate way of preventing human disease. At the same time, whatever we do will be passed on to generations to come, and it's extremely risky. Because if we screw up in some way and edit out the wrong thing, we have now impacted potentially millions of people. So when it comes to editing the genome of human embryos, the benefits are enormous, the risks are enormous, and that creates a volatile ethical debate.
Are there any policies that have been put in place to deal with some of these ethical issues?
So CRISPR technology is very, very new, but there have already been a few statements, policy statements, and groups that are thinking about how to deal with these challenges. In particular, in December 2015, there was an international summit in Washington, D.C., where scientists and ethicists and policymakers from all over the world came together to look at the challenges that CRISPR poses and to think of how to guide the use of the technology. And they came up with the following recommendations. For plants and animals, we can use the laws that we already have. For human beings, we should proceed with clinical trials for patients, and we should experiment on human embryos, but we should not engineer or edit embryos and start a pregnancy with them. So they drew the line at the level of actually using edited embryos to create children. They said, we're definitely not ready for this, but we are ready and we should encourage the use of CRISPR to try and treat patients and to try to edit embryos. The first trial in human embryos was approved in England, in the UK, just a couple of months ago. So it hasn't even started. Canada is in a unique position because we have a law from 2004 called the Assisted Human Reproduction Act that tells us that it is criminal in Canada to do research on embryos that would modify the embryo in a way that these changes might pass on to the children. And while we're not going to start a pregnancy with an edited embryo, Canadian legislation doesn't even let us do the research on human embryos. It plays it safe, yes, but it might create a situation where Canada is left behind in relation to other countries. So if other countries are starting now to do research with CRISPR on human embryos and Canadian researchers are not allowed to do this because the law says they can't, they might be left behind and they might start leaving Canada to go do their research elsewhere. That's also a risk. We don't want Canadian research to be left behind. So a number of bodies within Canada are now looking into changing maybe the law to allow the use of CRISPR in trials on human embryos.
Do you think that CRISPR is going to revolutionize medicine in the near future?
The media is very enthusiastic about CRISPR. It's been called the breakthrough of the year. Everybody thinks that the two ladies that discovered CRISPR-Cas9 are going to get a Nobel Prize. I agree with that. It's a safe prediction. Everybody thinks this is the next big thing in medicine. And maybe it is. I hope it is. For all those families with children or family members suffering from genetic disease that don't have a cure, I really hope this is it. This is the key to curing genetic disease. But we've been here before, and this is the phenomenon of hype. When we have a new technology, everybody gets excited, a little bit overexcited. The media wants to sell it to us as a magic solution that is just around the corner. We've been there with gene therapy, with stem cells, and we always find at the end that it's more complicated than we thought. It doesn't give the results we thought. It takes longer. Biology is very complex. So we end up running clinical trials for 10 years, 15 years, and we're making headway, but slowly. I believe that CRISPR will end up being like those other technologies. It will give solutions, but not tomorrow morning, and maybe not in the next 5 years. It will take a lot of tweaking, a lot of repetition of trials to make sure it's safe. We will probably try it on small groups, find out something's not working, try it on bigger groups. Using the word revolution gives the public the feeling that cures are available next year. And while we would love for this to be the case, it probably isn't. Biology is very complicated, and we need to move slowly and carefully so we don't hurt people while we're trying to help them. So my bottom line message is, yes, we should be enthusiastic. This is huge. This is exciting. This carries great potential, but it's very complicated, and it will probably take more time than the newspaper is telling us now.